Our next speaker is Kelly Schiabor, and she'll be speaking on investigating the molecular basis of species interactions in Saccharomyces cerevisiae mitochondria that are required for optimal attractiveness to Drosophila melanogaster. Thank you, Kelly. Thank you, guys. I'm really excited to talk to you today. So before you are pictures of the yeast, Saccharomyces cerevisiae, and the fruit fly Drosophila melanogaster. And both of these are our model species of molecular biology. And as I'm talking to a room full of fly biologists, we can all appreciate the fact that the fruit flies often escape our labs and find the yeast labs down the hall. And while this could be a laboratory oddity, it appears as though this is actually not coincidental. So both yeast and fruit fly actually also co-localize in nature, mostly on the surface of sugary substrates like fruits. And this co-localization is beneficial for both of these species. Further, interactions between organisms are very common in nature and likely shape the biology of these organisms in the way. But the problem is that most of these interactions happen in environments that are really hard to recapitulate in a laboratory setting. But this is not the case for melanogaster and cerevisiae. So studying the interaction between these organisms gives us a chance to study organismal interactions at the molecular level in a laboratory setting. So while this association is probably rich and has a lot of different aspects to it, I'm going to talk to you today about the most proximal piece of this interaction, namely, how do these two little species find each other in this big natural world? So just as the basics, Drosophila are going to most likely be using olfaction to find long-range, use long-range cues to find these yeasty substrates. And you know, those of us who have been out at night know that yeast actually produce a large array of volatile compounds as they ferment or metabolize the substrate. So the first question that I wanted to ask was, do Drosophila melanogaster prefer specific yeast strains over others based primarily on how they smell? And I reasoned that if I could figure out or find strains that differed in their preference, I could use yeast genetics to understand the molecular basis of these differences. So I went around to all the labs I could and found as many sequenced yeast strains as possible, and then developed a simple trap-based preference assay to measure this phenotype. So briefly what I did was, it's a large arena-based trap assay where I would bait the traps with different yeast strains, pairs of yeast strains, and then use the number of fledged traps in each trap as a way to quantify preference for strains. So I was screening across many different yeast strains, and the most surprising result actually came from when I baited the traps with two yeast strains, BY4741 or BY4742. So the flies consistently preferred BY4741 baited traps over traps baited with BY4742. So you can see all the trials here. The reason that this was so interesting is that I included these strains in my panel on a whim because they're actually the background strains for something called the yeast knockout collection. And these are two haploid cells that are actually isogenic to each other, except for at a couple of metabolic loci that we use to phenotype these strains in lab. So BY4741 is a methionine oxytrope and has gene deletion in the methionine biosynthesis pathway. And BY4742 is a lysine oxytrope and has a gene deletion in the lysine biosynthesis pathway. So here I had an example of a case where we saw a strong preference difference between these two strains, and they only varied at a discrete number of loci. So for that reason, I was able to do a very classic yeast genetic experiment. So what I did was I took these two haploid spores and crossed them to each other to form a diploid, and then spoilated this diploid back down into haploid spores. So this forms a tetrad. And this process effectively mixes up the genotypes of the parental strain, but within each tetrad, we're going to have 2-to-2 segregation of those varying traits. Therefore, what I can do is actually take the individual spores from within that tetrad and then test it against one of the parents. So I decided to test it against the unattractive parent, BY4742, and what we'd expect to see is a 2-to-2 segregation pattern in the behavior as well. And we should be able to correlate this behavioral pattern the genetic pattern to understand which genes were driving this preference. So I did this, and surprisingly, I did not see the segregation pattern as expected. So all 4 of the spores within the tetrad, the first tetrad I tested, were more attractive than 4-2. So they all looked like the 4-1 parent. So again, this is not that 2-2 segregation we were hoping for. But this is only one tetrad, so I did it again and again and again, and saw the same thing. So across all tetrads, no matter their genotype, all of the spores were more attractive than the 4-2 parent, or looked like 4-1. So it appeared as though this was not a Mendelian inheritance pattern that we were seeing, but it looked a lot like another inheritance pattern, namely a mitochondrial inheritance pattern. So a little bit of yeast biology for you guys. So yeast can actually survive without a mitochondria, and this is because they generally use the process of fermentation for energy metabolism. And so for this reason, yeast cells will actually just lose their mitochondrial genome from time to time. And these cells are called petite mutants. But what happens is when a petite mutant is crossed to a grand strain, a strain with a mitochondria, all of the resulting progeny will inherit that mitochondrial genome from the grandparent and will be grand themselves. So you can see that this actually matched the pattern that we were seeing a little bit better than the Mendelian one. It suggested that presence of a mitochondria and mitochondrial genome is somehow influencing fly preference. Okay, so I went back and sequenced our 2 parental strains and confirmed that indeed BY4742 was missing its mitochondrial genome and was a petite mutant. Oh shoot, sorry. All right, so the next thing I wanted to do was sort of confirm this independently in other strains. So the first thing I did was I went to another lab down the hall and got an independent BY4742 isolate with the mitochondrial genome, which I'm referring to here as BY4742G, and tested that against both 4-1 and 4-2P. So following this mitochondrial hypothesis, BY4742G was equally as attractive as BY4741 and was more attractive than BY4742P. I wanted to confirm that this had to do just with like a disruption of mitochondrial function and not just loss of the mitochondrial genome. So I went to the yeast knockout collection that these strains are a part of and retrieved a nuclear gene deletion strain that disrupts mitochondrial function and tested it against our 3 strains. And following the other results, this strain was less attractive than 4-1 and 4-2G and was equally as attractive as 4-2P. All right, so what have I told you here? It appears as though Drosophila and melanogaster prefer grand yeast strains, or strains with a functional mitochondria and a mitochondrial genome, over strains that don't have this. Again, if you remember, I was using a very basic trap preference assay, where the flies are actually just choosing between these baits based primarily on just how they smell. So if this is true, we hypothesize that presence of a mitochondria is somehow affecting the volatile profiles produced by these strains, and that the flies are able to use these differences in volatile cues as a way to tell these apart and go towards the ones they prefer. So to test this hypothesis, I used a technique called gas chromatography coupled to mass spectrometry, or GCMS. And briefly, what GCMS allows you to do is you can take the volatile headspace of a culture, break it down into its individual components, and then measure the amount of each of these components within the culture. So this produces something called a chromatogram, which you guys can see here. And you can use a chromatogram to sort of compare between cultures. So I did this first for our initial results. So BY4741 versus BY4742B. And this is what the average chromatograms look like for many GCMS samples. So as you can see, there are large differences in the volatile profiles produced by these 2 strains. And more specifically, BY4741 produces more and higher levels of many volatiles, which I've identified here with stars for you guys. And we hypothesize that these maybe represent potential fly attractants. Okay. So, yeast mitochondria influence both attractiveness and the volatile profile produced by a strain. So how could this be happening? So mitochondria are most famously associated with the process of respiration. So I wanted to first test the hypothesis of does respiration drive the production of these compounds and therefore is responsible for attractiveness? And so to do this, I took advantage of the dynamics of a yeast culture. So as I mentioned earlier, yeast actually prefer to ferment when there's enough glucose present in a culture. But as that glucose sort of goes away over time, they are forced to switch to the more energy efficient process of respiration. So given this time course, we would expect that if respiration was driving the production of these compounds and responsible for attractiveness, that a culture that had been grown for a longer amount of time would become more attractive than a younger culture and would produce more of our identified attractants. So I did this with our BY4741 strain. And what I found was actually the opposite. So the fermenting cultures were far more attractive than the later respiring ones. But this sort of matched what we saw in the GCMS profiles. So respiring cultures actually stopped producing many of the attractive compounds that we had identified in the comparison between BY4741 and BY4742P. Okay, so it doesn't seem like it's respiration that's responsible for the production of these compounds and attractiveness. So I moved on to sort of the next case. So although less appreciated, the mitochondria is also actively involved in nitrogen metabolism. And this sort of cued us to other experiments that I'd been doing at the time on how sort of nutrients affect the volatile compounds produced by a yeast strain. So when yeast are grown on lab media, they smell a lot different than when they're grown on fruit. And so there's a kind of an obvious and easy reason for this. Lab media is nutritionally essentially opposite of fruits. So lab media is very much glucose limited and has ample amounts of nitrogen, mostly in the form of molecular nitrogen. On the other hand, fruit media have high levels of glucose and limited amounts of nitrogen, and it's mostly in the form of amino acids. So you can imagine when growing on a more fruit-like media, there's going to be a high level of glucose, which is going to allow the cells to actively ferment, which we know is sort of the attractive stage. But the limited amount of nitrogen and types of nitrogen may force mitochondrial engagement. So to test if this hypothesis was true, I took a prototropic yeast strain and grew it on either lab media and fruit media and compared it with both behavior and GCMS profile. All right, so great news. Yeast grown on the prototropic yeast strain grown on the fruit media was far more attractive than the same strain grown on lab media. Further, this was associated with the highest levels of the attractants we had identified that separate 4-1 cultures from 4-2P cultures, as well as fermenting cultures from respiring ones. All right, so what have we learned? So it appears as though non-respiratory functions of the mitochondria influence the attractiveness of yeast to fruit flies, generally during the process of fermentation. And we think that this might be happening via the mitochondria's engagement in amino acid metabolism. And more generally, what have we learned? Fruit flies seem to be attracted to yeast grown on natural substrates. And natural substrates have a very unique nutritional composition, which drives the production of very specific volatile compounds that Drosophila have sort of evolved to sense. So with that, I'd like to thank my mentor, Mike Eisen, and the other members of my lab who are here and also presenting posters and have talks today. And yeah, with that, I'll take your guys' questions.
Great talk. I'm unclear. Did you grow them on different diets in a petite mutant where there is no mitochondria? Can you show that the volatiles are due to something other than the mitochondria? So you grew them on a strain that had a mitochondria, right? You grew them on the two different.
Oh, the two different media.
If you grew them on a petite strain, you could show that it's not due to mitochondria.
Yeah. So the problem. So I haven't done the experiment yet, but so our original petite mutant is an oxytroph. So it's actually really hard to combine like nitrogen metabolism and oxytrophy and all that kind of stuff. So I have run it on SC media, and it doesn't affect the volatile metabolism the same as when I grow 4-1 on either SC or YPD, which is kind of similar. So that's as close as I've gotten.
So you had two different haploids, one of which was petite, the other was normal. You call it grande. Did you ever look at the cross, the actual diploid? The diploid, which is functionally a third, not quite sex, does it have a different phenotype than either of the haploids that you were looking at?
So the diploid is equally as attractive as 4-1 and more attractive than 4-2.
And if you made tetraploids, would you call them venti?
Yeah.
Have you tested different strains of flies to see if they have different preferences, or will you test that?
Yeah, no, I have. I didn't have time to talk about it today, so this was all done in one Raleigh line, but there is variation in preference across lines, and we're working on it.
Did you separate male and female flies?
Yeah, so this is a combined mixed population, but males and females do have slight differences in their behavior. Females are more distinct in their preference, and males sort of seem to just follow along, honestly. Thanks, Nellie.